The topic that we shall be discussing is about autoimmunity. So, immune response, as the coin has two faces, similarly the immune response directed against the antigens or towards the host can be either acting in a protective role to eliminate the antigens from the body, or sometimes these immune response can cause a deleterious effect inside the human host, and these deleterious effect can be brought about either by the hypersensitivity or autoimmunity. So here we are going to discuss about what is autoimmunity and what are the different diseases related to autoimmunity. So the first and foremost thing that we should know over here is the self antigens. What are the self antigens, and how are these self antigens able to induce this autoimmune response? Self antigens are nothing but the antigens that belong to the human host, and when the self antigens are exposed to the immune system, these immune system produces antibody towards the self antigen, causing the destruction of the self antigen. And when is this possible? It is usually said that the lymphocytes that are produced during the fetal development are exposed to the self antigens or the antigens of the human host as self antigen, and there is no immune response that has to be produced against the self antigen. So that is what is called as immunological tolerance towards the self antigen. And when there is a breakage in this tolerance, you could see that the immune response going awry and producing antibodies against the self antigen, thereby destructing the self antigen, leading to different types of autoimmune diseases. So autoimmunity is nothing but the immune response that is developed against the self antigen is said to be called as autoimmunity, and it was Elix who has given this term as horror autotoxicus. So here in the case of autoimmune diseases or autoimmunity, instead of protecting self antigens, the immune response is directed towards the self antigen, thereby causing the damage. And this damage can either be a localized damage causing to certain specific organs in the body, or can be systemic and ultimately leading to the death of the person. So autoimmunity is immune response towards the self antigen. And how will you describe this autoimmunity? Is nothing but the structural and functional damage of the cells caused by the immunocompetent cells that are produced against these self antigens. So it is called as autoimmunity, and the diseases that are related to this is called are called as autoimmune diseases. And here you can see there is injuries to self. So instead of the organism that usually induces an immune response, here you can see the immune response developed against the self antigen without any antigenic stimulation. But sometimes it so happens that antigenic stimulation also induces these autoimmune diseases, and mostly it is possible that after the entry of the pathogen, there are many chances that it can lead to various types of autoimmune diseases or the disorders. And the most important thing here are the culprits, you can say, in inducing the autoimmune diseases are the autoantibodies. So these are called as autoantibodies because they act against the self antigen, and hence they are called as autoantibodies, and they are the one that usually induces this autoimmune reactions. So the first person who gave us a concept of this autoimmune disease is the Mechnikov, and Donat was the one who has established like these autoimmune diseases can be taking place in the humans too. And with the development of the Coombs test, we are able to find out the autoantibodies that can usually cause different types of hemolytic anemia. So ultimately, with the help of sensitive serological techniques, we are able to identify the autoantibodies that are directed against different types of organs or different types of self antigen in the body that induces the autoimmune diseases. So here we can find that the disease are related to the autoimmunity are usually because of the autoantibodies, and there are different types of autoantibodies that are mostly involved in developing these autoimmune diseases. Now coming to the features of autoimmune diseases, how will we know that a person is suffering from particular autoimmune disease? So first and foremost thing is that the person will express elevated levels of immunoglobulins. Mostly these are nothing but the autoantibodies that are in excess that you can see in this patients who are suffering from autoimmune diseases. Next thing is the demonstration of autoantibodies. This can be easily demonstrated with the help of various serological techniques that can be carried out in vitro to identify the autoantibody and the organ or the system that is usually involved in the autoimmune diseases. Next is the deposition of this immunoglobulin at the site of elections. So wherever there is a specific organ involved, you can see there is an enhanced production of autoantibodies, and there is a deposition of these autoantibodies at that particular site of election. So elevated levels of autoantibodies at the site of election can also be a feature of these autoimmune diseases. Next is the accumulation of lymphocytes and plasma cells towards the lesion. So this lesion is usually caused because of the inflammatory response that has been produced during an autoimmune response. So this autoimmune response develops the inflammatory response that helps in infiltration of the different cells of the immune system. So you can see the accumulation of all the different types of cells at that particular area, which is called as a lesion. Next thing is you can get benefit from immunosuppressive drugs and immunosuppressive therapy with the use of corticoids. So this can also be one of the parameters that can be used to know the feature of the autoimmune disease. Next thing is more than one type of autoimmune lesion is associated during an autoimmune disease. So it is not you can say that a specific organ is involved. Sometimes it can also become systemic if not treated properly. So more than one type of autoimmune lesion can be associated in an autoimmune disease, and it is a genetic predisposition. That is, it is usually seen in the persons who inherit from their parents. So it can it is not in all the normal individuals that you can see, but mostly you can see these type of autoimmune diseases, and it is a genetic predisposition. And here the most people that are affected with this autoimmune diseases are the females. So usually it is said that above 40 years age, most of the ladies or the females are able to get these type of autoimmune diseases, and these autoimmune diseases are highly chronic in nature, and if not treated properly can even lead to the death of the individual. Next is coming to the mechanism of autoimmunization. How is this autoimmunity usually developed in an individual, and what age and which are the factors that are responsible for the development of this autoimmunization? So the first and foremost is the theory that is proposed is called as a neoantigen. So this neoantigen is nothing but the antigen which is new, and the self antigen usually gets altered structurally or morphology due to various conditions such as physical, chemical or biological, and these self antigen can change or altered self antigens are called as a neoantigen. Now once this neoantigen is formed inside the body, the immune system cannot recognize this as self antigen, and they usually produce an immune response towards the neoantigen that are formed. So there are a variety of factors that usually develops the neoantigens in our body. So they are called as physical, chemical or biological factors that usually causes the alteration of the cell. Physical factors that are involved are usually irradiation. When we get exposed to UV rays, X-rays, etc., the cells usually changes the morphological characters, thereby changing into a neoantigen. And sometimes photosensitivity and cold allergy can also cause the cells to get altered to form a neoantigen. Next are the chemicals that are mostly involved are the drugs. That is, the drugs are nothing but the antibiotics, or sometimes some people are prone to certain habit of taking drugs. So these drugs when introduced into the body can convert the normal self or the self antigen to the neoantigen. And contact dermatitis, which is a type 4 hypersensitivity reaction, can also lead to autoimmune disorder, because here the skin proteins are mostly involved. Skin protein, when mixed with certain type of drugs used in the cosmetics, can cause a change in the structure of the skin cells towards which the immune response can be developed or produced, leading to contact dermatitis. So here you can say it is antibody mediated type 4 hypersensitivity or autoimmune disorder. And most of the drugs that usually are injected can cause this type of autoimmune disease, which are called as the anemias. Anemia is nothing but decrease in the number of the RBC. RBCs. Now here, the drugs such as penicillin, what they do is, in some allergic individual, they go and bind to the RBCs and they completely change the morphological character of the RBCs. Now this neoantigen, which is formed by the penicillin and RBCs, usually are taken as a non-self antigen, and towards this non-self antigens the immune response is developed, and the RBCs are usually eliminated in combination with the complement mediated lysis, thereby decreasing the number of the RBCs, leading to anemia. So it is called as a drug induced anemia. Next are the biological characters, or the biological factors, which are nothing but the organisms. Now when the organism gets introduced into the body, they have the ability to modify the structure of the cell, mostly in case of viruses. When the viruses are known to be pathogenic, when they go and enter into the host cell, the pathogenic character of the virus is by making its DNA or the genetic material enter into the cell. Now when the genetic material enters into the cell, there is a lytic cycle or the lysogenic cycle that is usually produced, and this causes a change or the alteration in the morphological characters of the cell. Now these are called as the virus infected cells. So towards this virus infected cells, the immune response can be produced in autoimmune disorders. So here biological factors are nothing but the infectious microorganisms such as the viruses, and infectious mononucleosis, which is usually caused by the organism, is also a type, is said to be preceded by the autoimmune diseases, and the certain enzymes released by the organism exposes the T antigens. Now these T antigens usually are the one that develops autoimmune diseases, but they are not as harmful as the other autoimmune disorders, and sometimes mutation can take place via physical mutagens or the chemical mutagens in a cell, thereby causing a change in the characteristics of the cell, making it a non-self, towards which the immune response can be developed. So neoantigens are nothing but the self antigens that have been altered physically and chemically, so that the self antigen becomes a non-self to the immune system, and the immune response that is developed against this immune, that is non-self cells, and thereby causing the destruction and leading to autoimmune diseases. Next important mechanism that was proposed was thought of cross-reacting foreign antigens. So cross-reaction is a phenomena where when an antigen enters into the human host, the antibodies are produced against it. But because of the antigenic similarity between the organism and certain components of the human host, the antibody, instead of going and reacting with the organism, they go and react with the self component, thereby causing a damage to the self component. So this usually happens when there is an antigenic similarity between the two different types of antigens. So here the cross reaction is a very rare phenomena, but it usually occurs, and this could also lead to autoimmune disorder. So there are certain organ specific antigen that are specific towards a particular species. Now when these organ specific antigen, when they are introduced, they can induce the production of antibodies, and which can go and destruct the self cells. So the best example is the neurological disability after the neural vaccine is given in a person. So the neural vaccine is usually prepared from the sheep's brain that is usually infected. Now when this neural vaccine is given, the antibodies are developed against the that is the neural vaccine, but instead of going and acting on it, they go and act on the neurological characters of our body, thereby causing the neurological disorders in the body. So that is one example of the cross reaction. Next is the streptococcal M protein. The organism Streptococcus has an M protein on its surface, which has exact antigenic similarity with the heart muscle. Antibodies that are produced against the streptococcal M protein, instead of going and acting towards it, they go and act the heart muscles and damage the heart muscle. And next is the nephrogenic strains of streptococci. They share the antigenic similarity with the renal glomeruli antibodies, or the autoantibodies you can say. They destruct the renal glomeruli of the kidneys. Next is molecular mimicry. That is another proposed concept for developing autoimmune diseases. Molecular memory is nothing but when these again antigen and the self-antigens share the same antigenic character, but here they have identical peptide sequence. In the case of cross reaction, they share identical antigenic character, but they are not very similar to each other. But here in the case of molecular memory, there is an identical peptide sequence that is specific both towards the antigen, that is the infectious microorganism, and the self-antigen. So the autoantibodies are produced against the self-antigen because of the identical peptide sequence that is shared by both the infectious microorganism and the self-antigen. Example is arthritogenic Shigella flexneri and HLA-B27. Next is Mycobacterium tuberculosis and joint membrane. They have identical peptide sequence. So autoantibodies, instead of going and acting upon the Mycobacterium tuberculosis, they go and destruct the joint membrane. Next is Coxsackie B virus and myocardium. Next is the best example that can be given over here about the molecular mimicry is of the infection with the HIV-1 virus. So HIV-1 virus has a structural component which is called as a glycoprotein 120, and the autoantibodies that are produced against this, they instead of going and reacting with the HIV organism or the GP120, they go and act upon the astrocytes of the nervous system, which are nothing but the cells of the central nervous system, and they completely causes the damage to the central nervous system, leading to the central nervous system disorders in the human body. So here the apoptosis of the astrocytoma cells usually takes place because they target the V3 like protein that are present in the astrocytes. So usually it is said that HIV infections usually causes the most of the central nervous disorders because of the production of autoantibodies in case of autoimmune diseases. Next concept that is proposed is a polyclonal B cell activation. So whenever an antigen enters, the most important characteristic feature of the immune response is the production of specific antibody towards a particular antigen. So here we have an antigen that is gaining an entry into the human body, and it has certain epitopes which are of certain structure. Now towards this structural epitope, the antibodies are produced, and they go and bind based on their complementary base pairing. So that is what is called as specificity. But here in case of autoimmune diseases, there is a polyclonal B cell activation. Instead of one antibody that being produced against that particular antigen, more amount of antibodies of non-specific concept are usually produced. So these can be induced mostly by different type of factors such as chemical. Example of chemicals that can induce this polyclonal B cell activation is the 2-mercaptoethanol. You have bacterial invasions usually having PPDs and LPS, that is lipopolysaccharide layers. They usually produce these polyclonal B cell activation, and enzymes such as trypsin. You have antibiotics, nystatin, and infection with the mycoplasma, and viruses, EB virus, and the parasites such as malaria. So these polyclonal activation is nothing but production of more than one type of antibody towards the different antigens, and the production of excessive amount of polyclonal antibodies can also lead to autoimmune diseases. Next concept that is proposed is of immunological tolerance. Now tolerance is nothing but whenever a self antigen is exposed to the lymphocyte during the fetal development, the immune response is not directed towards the self antigen. So it is said to be tolerated against the self antigen. It usually happens during the fetal development, where the self antigens are usually exposed to the lymphocytes and no immunological response is developed. But once these tolerance is lost, then it can lead to autoimmune diseases. So even even though the antibodies or the leukocytes or the lymphocytes that are produced against the cells, they will be eliminated by clonal selection during the selection process. Now, because of this clonal selection, you cannot see the production of self-reacting antibodies or the autoantibodies. But once the tolerance is lost, then you could see the production of the autoantibodies that are directed toward the self-antigen. So these are called as forbidden clones. So once the tolerance is lost, the forbidden clones are able to produce the autoantibodies that can be directed against the self-antigens. Next are the T cells. It is not that only the antibodies that are involved in autoimmune diseases, but also cell-mediated immunity brought about by the T cell can also be involved in the development of autoimmune disorders. So it can be said that the autoimmune diseases can be caused both by the humoral immunity as well as the cell-mediated immunity. So here, in the case of autoimmune disorders which are brought about by the cells, here you can see there is an enhanced T cells and decreased T suppressor cells. It is said that these T suppressor cells are the one that maintains the regulation of the production of T cells in the T cells or the other cells in the human host. So once there is a decreased T suppressor cell, there is no specificity that can be seen between the antigens and the antibodies, and more amount of immune response is developed towards the self antigen or the antigen that is coming from the or as a foreign antigen. So here the T cells that are excessively produced are the T-H cells, and the T-H cells are the one that produces the cytokines to activate the other cells of the immune system. So there is a decrease in the production of TS cell, as well as there is an inactivation of the macrophages, and ultimately leading to the autoimmune disorder. So here you can say the defect is in the thymus, where excessive production of the TH cell and decreased TS cells are usually seen in developing the autoimmune disorders. Now next concept is the sequestered antigens. Sequestered antigens are nothing but the hidden antigens. So these antigens are of two types, you can say. One best example of the sequestered or the hidden antigen is a lens protein that is usually enclosed in a capsule. Now when there is an injury to the capsule, these antigen will be exposed, towards which the immune response can be developed by producing the autoantibodies. So the lens antigen of the eye sometimes can be exposed due to the injury and can cause the development of immune response towards the eye. Next example is a sequestration in time. That is the best example here we can discuss is about the production of the sperms. The sperms are usually produced after the puberty. Now this has not been exposed to the lymphocytes in the initial stages of the life. So now the production of the sperms can lead to the development of autoantibodies against these spermatozoa or the sperm producing cells, and can cause a damage, which is the reason why the arthritis is seen in the persons, and which results later in the formation of the mumps or the causation of the mumps. So that is what is called as sequestration in time. That is hidden antigen that is exposed during the later part of the life. So next proposed concept of autoimmune disorder is a defect in idiotypic and anti-idiotypic networks. So in order to develop an immune response, the genetic character is very important. The genes that are involved are very important. So there is a defect in the genes that produces the immunological response. So the gene that is involved is gene IR or immunoglobulin genes. So when there is a defect in these genes, you could see the production of autoantibodies that goes and interacts with the self antigen. So the proposed concept over here is a defect in the genetic character or the genetic basis of the individual. So this can also be taken into account as a genetic predisposition, where this character is inherited by the generations and generation together, who could see the formation of autoimmune disorders in the humans. Next is how do we classify these autoimmune diseases, because these autoimmune diseases not limited to only one particular part of the body. It can be seen throughout the whole part, and it can affect different components or the different organs or the different tissues in the body. So based upon the lesion that is formed, based upon the mechanism, we can classify the autoimmune diseases into four types. One is hemocytolytic autoimmune diseases, where the targets are the RBCs, and the autoantibodies antibodies are produced against the RBCs, leading to anemias, different types of anemia. Next is localized or organ specific, where a specific organ is usually targeted and the autoantibodies are produced against that particular specific organ. Next is systemic or non-organ specific. Here the antibodies are not produced against a specific antigen, but towards different self antigen that are distributed throughout the whole body, and the autoantibodies can interact at the same time towards a different part of the body and can lead to systemic autoimmune diseases. So it is a non-organ specific. Next is a transitory disease. These transitory diseases are usually seen only when the person is introduced with certain drugs, and when the drug is present, it usually enhances the autoimmune disorders, and once the drug is withdrawn, then you can see the person coming back to his normal health. So it is only for a temporary period of time till the drug is present inside the human host. So this is how you can classify the autoimmune diseases. Let us see each of them in details. First, let us deal with the hemocytolytic autoimmune diseases. As told, the target cells are nothing but the RBCs, and the autoantibodies are produced against these RBCs. Now, based upon this, two types of anemia can be classified, and two types of autoantibodies are usually seen in the case of hemolytic anemia. So one is called as a cold autoantibody, the other one is a hot or the warm autoantibodies. Coming to the cold autoantibody, it was proposed by Donath in paroxysmal hemoglobinuria patients, and the autoantibodies are said to be complete and mostly of IgM type, and they can interact or cause agglutination at only 4 degrees centigrade and cannot show reaction at 37 degrees centigrade. Hence, they are called as cold autoantibodies. Seen in paroxysmal cold hemoglobinuria patients, and they usually are manifestations of, you can say, primary atypical pneumonia, which is caused by mycoplasma, trypanosomiasis, and black water fever. Now coming to the warm autoantibodies, they can interact at 37 degrees centigrade, and hence they are called as warm autoantibodies. They are said to be incomplete and they are non-agglutinating. So in order to identify these type of antibodies, you need to go with the direct Coombs test. Coombs test is the one that is helpful for us in identifying these type of autoantibodies. Next is these are usually induced by the drugs such as sulfonamides, that is the people who are on sulfonamides, antibiotics, and alpha methyl dopa. So these are the one that are targeted against the RBCs. So they go and bind to the RBCs and they cause the destruction of the RBCs by complement mediated lysis, leading to anemia or a reduction in the number of the RBCs from the body. Now next is autoimmune thrombocytopenia. So here in the above disease we have dealt the target to be the RBCs. Here the target in the thrombocytopenia are the platelet cells. So autoantibodies are produced against the platelet cells, and the disorder that is associated is called as a idiopathic thrombocytopenia purpura. And if the autoantibodies are produced against the drug induced, like when the drug is injected, they go and bind to the platelet and they causes a change in the conformation of the platelet, thereby enhancing the production of autoantibodies. So mostly seen in serorbit purpura, and here it is called as antibody mediated hypersensitivity reaction, because the antibody is going and binding to the platelets that have been changed by the drug. Next is autoimmune leukopenia. Here the autoantibodies are produced towards wide variety of the organs of the body, mostly seen in the type 3 hypersensitivity reaction, which is induced by the antigen-antibody complex, called as systemic lupus erythematosus and rheumatoid arthritis. So type 3 hypersensitivity can also be mediated and can lead to autohemolytic or autoimmune leukopenia. Next is organ specific. So here it is the hemolytic or the hemocytolytic autoantibodies that are produced against the RBCs. Next, coming to the organ specific, where a specific organ is usually targeted by the autoantibodies. So the target organ is in case of Hashimoto's thyroiditis or Hashimoto's disease, also called as lymphadenoid goiter. So here the target organ is the thyroid, and the autoantibodies are produced against the the variety of antigens that are present on the thyroid organ. So specifically they are produced against the thyroglobulin receptors that are present on the thyroid. So the autoantibodies are called as anti-thyroglobulin antibodies, mostly seen in the females, and here there is an enlargement of the thyroid gland. There is a replacement of the normal structure with the infiltration of the lymphocytes, plasma cells, and a variety of other immune cells. So hypothyroidism is one of the symptoms that can be seen in Hashimoto thyroiditis, and enlargement of the gland that is seen because of the infiltration of the gland with the help of different cells of the immune system, and it is forming a lymphoid tissue because the presence of more amount of lymphocytes, plasma cells, and the other cells that are belonging to the immune response. So here the target organ is the thyroid. Autoantibodies are the anti-thyroglobulin antibodies and other antibodies that are produced against a wide variety of antigen, and the symptoms that can be seen is the hypothyroidism. Next is thyrotoxicus, also called as a Graves' disease. Again the target organ is the thyroid organ, and the antibodies are specifically directed against the thyroglobulin, and here also you can see the lymphocyte infiltration. The antibodies are a long-acting thyroid stimulator. These are the IgG antibodies that are produced during an immune response towards this Graves' disease. It is the antibodies that go and bind to the hormone receptor and makes it excessively secrete that hormone, which is called as thyroid hormone, and it is leading to hyperthyroidism. So in the previous case it is hypothyroidism. Less amount of thyroid hormones are usually produced, whereas in this case, because the antibodies goes and binds to the receptor and secretes more amount of the hormone, excessive hormone, it is called as a long acting thyroid stimulator, secrete excessive hormones of thyroid origin. Next is the Addison's disease. Addison's disease is nothing but here the target organ is the adrenal gland, and the autoantibodies are produced against the zone glomerulosa, causing the infiltration, again decrease in the production or decrease in the normal functioning of the adrenal gland, leading to Addison's disease. Next is autoimmune orchitis, that is already dealt with, where the autoantibodies are usually produced against the sperma or germal epithelium and aspermatogenesis. And here you can see the lymphocyte infiltrations of the testis, and the antibodies that are produced destructs the sperms or the germinal cells, leading to orchitis and followed by the mumps. Now next is the myasthenia gravis. Here you can see the autoantibodies produced against the myoneural junction. So there is a malfunction of the myoneural junction, and the target over here is the acetylcholine receptor. Now this acetylcholine receptor is very important in maintaining the contraction and relaxation of the muscles. So once these acetylcholine receptors are damaged or they are blocked, they will not be able to carry out the normal functioning of muscle contraction or the relaxation. So here the target organ is an acetylcholine receptor, which like the autoantibodies completely impairs the muscular function, leading to the contraction of the muscle. So here the thymus in this area is usually can be seen as a lymphoid hyperplasia with more amount of germinal centers. The best example that can be given about the myasthenia gravis, which is a pathogenic respiratory disorder, it's seen in autoimmune patients. So the effect of MuSK autoantibodies, muscle specific kinases autoantibodies. So these muscle specific kinase autoantibodies are Ig4 type of antibodies that are produced against this MuSK cells. So here what they do is they go and bind to the muscle and inhibits the LRP4 from binding to it, and agrin mediated function is completely lost, thereby causing this particular type of disease. So effect of MuSK autoantibodies destructs the normal functioning of the kinases that helps in the relaxation and the contraction of the muscles. Next, coming to the autoimmune diseases of the eye, here as already discussed, the lens is an important component of the eye which is enclosed in a capsule. Now once when the lens get exposed due to certain types of injury, this can lead to the immune response being targeted towards the lens protein. So after the cataract surgery, there is an immune response that is directed against the eye, which is called as phacoanaphylaxis. So like after the surgery, you can see the autoimmune diseases that can take place in the person who had undergone a cataract surgery, and this is called as the phacoanaphylaxis. Next is sympathetic ophthalmia, where there is a perforating injury to the iris or the ciliary bodies, and leading to this type of condition, which is called as a sympathetic ophthalmia in the other eye. It is not in the eye where the destruction is taking place, but it is directed towards the other eye. So it is these are the two important autoimmune diseases that are directed towards the eye. Next is the pernicious anemia. Pernicious anemia is included over here in the organ specific because there are certain specific cells that are targeted by the autoantibodies. So here the autoantibodies are directed against two important components. One is the parietal cells which are present in the gastric mucosa and leading to achlorhydria and atrophic gastritis. So destruction of these parietal cells by the autoantibodies can lead to achlorhydria and atrophic gastritis. Next is the autoantibodies that are produced against the intrinsic factors. So this intrinsic factor is usually seen in the small intestine, and this is the one that helps in the absorption of the vitamin B12. Now this vitamin B12 is very essential for the production of the RBCs, or you can say at hematopoiesis. So destruction to this intrinsic factor can lead to prevention of the absorption of the vitamin B12, and thereby leading to anemia, because RBCs cannot be formed with the help of this, without the help of this vitamin B12. So in this case of pernicious anemia, the patients are usually treated with the injection with vitamin B12. However, next is the autoimmune diseases in nervous system. You can say here there is destruction of the autoantibodies that are directed against the nervous system, which is very fatal and can lead to the death of the person and completely damages the central nervous system of the affected patient. So here the first example that we'll be dealing is a neuroparalytic accidents that usually takes place following the rabies vaccination. So the rabies vaccination is called as a neural vaccine prepared from the sheep brain that has been affected with the rabies or the virus. So they are made as the vaccine and given to the person. So the antibodies that are developed against this can go and interact with the nervous system and causes the neurological injury to the human host, causing the fatal diseases of autoimmunes. Next is Guillain-Barré syndrome. Here the target organ is the peripheral nervous system. Again, the same complication that is associated is the destruction to the central nervous system. Now coming to the autoimmune diseases of the skin, the autoantibodies can be generated against the different components of the skin. So the intracellular cement substances are the one that binds the different cells of the epidermis. So here the autoantibodies are directed towards the intracellular cement substances causing pemphigus vulgaris. Next is towards dermal epithelial junctions, autoantibodies can be generated leading to the bullous pemphigold, and dermatitis herpetiformis is the one that where we could not see any type of specific autoantibodies that is related, but there are theories that is coming forward to say that this dermatitis herpetiformis can also be an autoimmune disorder. So these are the different autoimmune disorders that are associated with the specific organs. Now coming to the systemic or the non-organ specific autoimmune disorders. So there is immune response that can be generated against the different antigens, the self-antigen that are distributed throughout the whole body, and the immune response can interact with a variety of self-antigen and causes systemic diseases. So here it is said that it is a disease of connective tissue lesion. And the best example here that can be studied are systemic lupus erythematosus, rheumatoid arthritis, polyarthritis nodosa, Jorgen syndrome, and dermatomyositis and scleroderma. Coming to the systemic lupus erythematosus, here it is a chronic autoimmune disorder because it is involving the whole body and the different self-antigens that are present in the human body. And the autoantibodies are produced against different cells such as cell nuclei, intracytoplasmic cells, immunoglobulins, thyroid cells, etc. And there is a loss of immunological tolerance that can be seen in case of this systemic autoimmune diseases. And the most important factor that can be recognized during this systemic lupus erythematosus is the LE cell phenomena. LE cell phenomena is nothing but an LE body which is a neutrophil, and this neutrophil is the one that has damaged nuclei, and it consists of a large number of or you can see a large shape of the cell that is completely surrounding the neutrophil inside, and there is a damaged nucleus that is associated with LE cells. So it is an immunological damaged nucleus which is nothing but a neutrophil, and this can be seen inside a neutrophil or can be seen outside the neutrophil in the plasma or the blood. And the neutrophil, when engulfs these type of nucleus, they form the LE body, and you can detect or demonstrate this LE cell with the help of various serological techniques, mostly the anti-DNA antibody test, anti-nuclear DNA antibody test. Loss of tolerance or the immunological tolerance can be best studied in case of systemic lupus erythematosus, where the macrophages usually in a normal person is able to bring about the apoptosis, and the cell don't become a secondary signal and don't release the danger signals, and there is no accessibility to the self antigen due to the apoptosis that is usually caused by the macrophages. But here in case of systemic lupus erythematosus, the macrophages loses its function to carry out the apoptosis. Danger signals are released by the secondary necrosis, thereby the self antigens or the autoantigens are expressed, leading to the inflammatory response. So this is how the immunological tolerance is usually lost in autoimmune disorders, and as discussed, they can be easily detected with the help of anti-nuclear antibodies and identification of the LE cell under the micrograph. Next is important type of systemic disorder of autoimmune diseases are the rheumatoid arthritis. So here there is a systemic or symmetric polyarthritis, muscle wasting, and subcutaneous nodules that can be seen in this rheumatoid arthritic patient, and it is because of the immune complex that is formed, and towards the immune complex the autoantibodies are produced. They usually go and try to extract these immune complexes that are deposited in different parts, and these immune complexes, mostly in case of rheumatoid arthritic patient, they go and hide themselves in the synovial junctions. So while extracting these immune complexes by the normal immune response, what they do is they completely damage the synovial joints, leading to the rheumatoid arthritis. So antibodies that are produced are the autoantibodies, which are called as the rheumatoid factor or RH antibody, rheumatoid factor, RA factor, sorry, and it is leading to serositis, myocarditis, and vasculitis, mostly seen in the women again above 40 years of age, and the target here is the synovial membrane, and a rheumatoid factor can be seen associated with this, and it is nothing but an autoantibody against the antibody, that is the IgG antibody. Now this IgG antibody is the one that is involved in the formation of an immune complex whenever an antigen enters. So these immune complexes that are formed with the antigen and antibody, towards this IgG antibody, autoantibodies will be produced, which are called as the rheumatoid factors. And the best test to identify these rheumatoid factor is the agglutination test. Example is a Rose-Waller test. Now coming to the another one, which is called as a polyarteritis nodosa. The target organs are the arteries. Necrotizing angiitis of the arteries can be seen in this disorder. Coronary thrombosis, cerebral hemorrhage, and gastrointestinal bleeding are the symptoms of this disease, and it can lead is usually caused said to be the reason for this is a serum sickness, and the immune complexes of the viruses such as hepatitis B virus antigen can also induce the formation of this autoimmune disorder, which is called as a polyarteritis nodosa. So target organ is the arteries. Next is Jorgen syndrome, is a triad of conjunctivitis associated with the sicca, that is a dryness of the mouth, and can also cause due to the rheumatoid arthritis, and it is said to be a collagen disease because it is involving a connective tissue, and this can be detected again with the help of anti-nuclear antibodies. Next is the transitory autoimmune process. As already discussed, it is a drug-induced autoimmune disorder, and it is mostly seen in the patient who are on drug therapy. So the person who are allergic towards certain drugs can sometimes cause this autoimmune, lead to autoimmune disorders. So drug therapy that is leading to anemia, thrombocytopenia, and nephritis. And here you can see when the drug enters, it causes the antigenic alteration of the cells, leading to autoimmune disorders. Now once the drug is withdrawn, you can see the person coming back to his normal health. So it is a transient alteration, you can say. It is not a permanent alteration leading to autoimmune disease, but once the drug is withdrawn or any thing that is associated with transitory autoimmune disease is withdrawn, the person becomes normal again. So these are the different autoimmune disorders, that is the immune response that can be directed towards the self-antigen and causing a damage to the self-antigen, leading to localized, or it can lead to systemic autoimmune disorders, ultimately leading to the death of the person.